Dear participants, welcome to this international conference on microalgae, biodiversity and bioprospecting. Dear participants, congratulations and all the best for the successful presentation of your papers. We are excited to have one by one with your presentation. Will be in 10 minutes for each participant. So the presentation will be in 7 minutes and followed by 3 minutes discussion. Hope you all get the schedule. So if anybody absent, next person will be called. So all of you get ready with your work. First of all, I call Bashida Banu for kindly present your work. Bashida Banu, are you there?
Bashida Banu, you're requested to turn on your mic.
Bashida Banu, kindly switch on your mic.
Bashida Banu, can you hear us? It seems like Bashida Banu is missing from the group. So I'm inviting the next person, Ms. G. Rama Prabhavati, Research Scholar, Department of Agronomy, Annamalai University. You can start your presentation. Ms. Rama, are you there? Next presenter, Ms. S. Swati, Research Scholar of Agronomy Department, Annamalai University. Are you there? I invite Ms. S. Swati, Research Scholar of Agronomy of Annamala University, to start her presentation. Ms. Swati,
Ma'am, yes, ma'am.
can you start your presentation?
Yes, ma'am. Ma'am, wait a minute. I'll share my slide.
Swati,
Ma'am, wait a
we
minute.
are not able to hear your voice.
Am I audible, ma'am?
Yeah, now audible. Present your slide first.
Ma'am, wait a minute, ma'am. I will share my slide. Hello, ma'am.
Yes, Ms. Swati.
Ma'am,
No,
I
you have
can't.
not yet shared your screen.
Ma'am, I can't be able to share my slide. Only the host can share in the meeting.
No, I've already made you as a co-host.
No, ma'am, I can't be able to share.
I'm giving this third line. Swati, can you please try once again?
Yes, ma'am. Only host connection. The host of American. Ma'am, please invite another person, ma'am. After 10 minutes, I will join, ma'am.
Okay, Swati. And now I invite Ms. Bashida Banu, PhD scholar in botany from Holy Cross College, Nagarcoil, to present her presentation.
Okay, ma'am.
Okay. I just want you to remind that you have been allotted 10 minutes time, on which 7 minutes you can use for your presentation, which will be followed by 3 minutes discussion. Is it clear?
Okay, ma'am.
Okay, you can proceed.
Ma'am, there is some error in my file, ma'am. Will you give 10 more minutes? Please invite another person.
Okay.
May I invite Rama Parvati? Rama Parvati, are you here?
Ma'am, yes, ma'am.
Now I invite Ms. G. Rama Parvati, Research Scholar, Department of Agronomy, Annamalai University, to present her paper.
Dear participants, kindly let me know whoever is willing to present can present first. You can raise your hand. Raise your hand in your control panel, please.
Hello. Good afternoon, madam. Madam,
Sir,
I am
your
audible.
voice is not audible, sir.
Madam, whether I am audible now?
Yes, sir. Kindly let me know your name, please.
Madam, I am Dr. Sabrinathan, Associate Professor Head, Agriculture College Research Institute, Madurai. I was scheduled to present my presentation tomorrow, but I was asked by the organizer to present today. Is that okay?
Sir, your voice is not audible.
Is it audible, madam? Now? Hello.
Yes, sir, now audible.
Okay. Shall I present now? Hello.
Sir, your name?
Dr. K.G. Sabrinathan.
Sabrina. Sir, if
Yes,
you are
sir.
ready, you can present now.
Okay. I was disabled I think you need to enable me to share my presentation.
Wait a minute, sir. Sir, now you can share your slide.
Is it visible, madam?
No, sir. Yeah, it's visible now.
Okay. Good afternoon to all of you. I am Dr. K.G. Sabrinathan, Associate Professor Head Microbiology of Agricultural College Research Institute, Madurai. I am presenting my paper on extraction and characterization of food-grade phycocyanin pigment from freshwater cyanobacteria. As that pigmentation or the coloring of the food is always preferable by the consumers. Nowadays, the food preparation people are using many. I am audible, madam? Is it audible?
Yes, sir, you are audible.
Okay. So the food producers, are the people who are preparing food are adding a lot of artificial colorants, which is highly dangerous to the human beings. So it is always there is a search for a organic food colorants. We have tried the naturally available phycocyanin pigment from the freshwater cyanobacteria as a natural food color. As we know, the cyanobacteria, blue green algae are prokaryotic photoautotrophs, which has been already proved as a biofertilizer. So it is a very good biofertilizer which is used in the rice field. We have tried to use this cyanobacteria to extract the biopigment, to use the colorant of the foods like cake, jam, jelly, we have prepared. Because these cyanobacteria is known for its amino acid production, fatty acids, as well as many antibiotic-like substances production. So in cyanobacteria, there are different phycobiliproteins like phycoerythrin, phycocyanin, and many phycobiliproteins are present. So we can extract this pigment from the cyanobacteria and use as a food colorant, which is a protein as well as safe for the consumers. So in the present study, we have standardized, we have screened different cyanobacterial strains for the production of phycocyanin. When we talk about the phycocyanin, it is a very good protein which is having anti-diabetic, anti-cholesterol, immunomodulatory effect. So in addition to the coloring, the consumer will get all the medical benefits from this pigment. So we have tried to, we have screened different cyanobacterial cultures, as well as we have purified this pigment, and we have evaluated the toxicity of this pigment in the silkworm. And we have finally prepared different food products with this pigment. So this is a pigment which we have isolated. You can see the different shades of phycocyanin. So based on the intensity or the extraction method of the phycocyanin extraction from the cyanobacteria, the shade of the color will differ. Because normally we used to get this blue color from the cyanobacteria. So these can be used as a food colorant to add any type of bakery based foods or any type of baked foods or any food material. And as well as it is a very good protein for the consumer. So we have screened, how we have screened means we have grown the cyanobacteria in the BG level medium, which is a unique medium for cyanobacteria. And we have grown these cyanobacteria in a illumination chamber at 28 degrees centigrade with 3000 lux intensity, because as cyanobacteria is a photosynthetic organism, the light intensity is more important. We have a exclusive light chamber where we have grown this cyanobacteria by periodical shaking also. Finally, after 3 weeks, we have evaluated the phycobiliproteins content, with the help of the Bennett and Bogart methodology. So these are the cyanobacterial cultures which we have used for our study. This is a big cell. Whether I am audible?
Yes, sir.
Okay. So this is the big cell which you are seeing here is a heterocyst, which is used by the cyanobacteria for the nitrogen fixation. Here you can see the big cell. So this Vestalopsis, Anabina, Nostoc, Oscillatoria are the exclusive culture. We have a huge culture collection, which has been collected from the rice ecosystem, which has been used as a biofertilizer as well as to extract the fine compound from the cyanobacterial biomass. So in addition to the fine like antibiotic-like substances, medical-related substances, as well as these food colorants, many materials can be extracted from these cyanobacteria-like materials. And also the growing the cyanobacteria is very easy as India is gifted with huge amount of sunlight. So we need only fresh water and enough sunlight to grow the cyanobacteria in mass level. So this is a screening of the cyanobacteria. So we have screened different cyanobacterial cultures and we found Vestalopsis. We have the culture called Vestalopsis, which produce huge amount of phycocyanin, like 32 microgram per milligram of dry weight. So after growing of the cyanobacteria in the B11 medium, we used to collect the cell pellet and we used to collect, we used to extract this phycocyanin by the freeze drying method. We have standardized a method called freeze drying method. Here you can see the result product. This is a phycocyanin, which can be used as a colorant, as well as nowadays they are using even for tagging different proteins. This phycocyanin is used as tagging for different proteins, as well as it can be a feed additive for many animals as well as birds to increase its immunomodulation, as well as it was having a very good antibiotic-like substances to kill the pathogenic organisms. So it can also be used in the plant system as well as animal system. To conclude this, we have screened 8 cyanobacterial cultures and we have found this Vestalopsis genus produces huge amount of phycocyanin, and we have also evaluated the stability of the phycocyanin with different temperature, light and pH, and we have found the very good stability of the phycocyanin which we have extracted, and we have evaluated the protein content of the phycocyanin pigment, and we found the protein content is 10 to 13 milligram per ml of the pigment, and we have also performed HPLC analysis of the phycocyanin to evaluate the purity of the phycocyanin, and we performed the STS page analysis to evaluate the molecular weight of phycocyanin. We have found 38 kilodaltons, which is exclusively a pure phycocyanin. Coming to my second research paper on development and evaluation of stress-tolerant cyanobacterial consortia extract spray to various sodicity level in rice ecosystem. As you know, after the green revolution, we are using more amount of chemical fertilizers to the farming system. These huge amount of chemicals as well as fertilizers leaves the soil into different pH problems like sodic soil, acidic soil, saline soil, as well as the seawater intrusion. The seawater, as the seawater level is increasing due to global warming, the seawater intrusion, the seawater is coming into the place where the peoples are living, habitat, or into the agricultural land. So the soil is getting sodicity. So to remove this sodicity, this algae can be used as a efficient tool. So this algae can hold the sodium ions and it can relieve the soil from the sodic problem or sodic injury. If we grow the plant along with this sodic problem, the plant will be injured or the growth will be stunned. The plant will not give enough yield. So there is a necessary to remove the sodicity problem from the soil. So the cyanobacteria can be used as an excellent tool to remove the sodicity or acidity or alkalinity in the agricultural ecosystem. So as we know, the blue-green algae is found in almost in all the environment. It can be seen in the freeze environment. It can be in the higher temperature also, in the mesophilic temperature, starting from the ocean to fresh water. It can be found everywhere. Even if there is a moisture in a wall for a long time, you can see the growth of algae. So this blue-green algae is well-known organism for to fix atmospheric nitrogen. Because as we know, we are using urea as a nitrogen source in the rice ecosystem, which is produced by the Haber-Bosch process, which needs huge amount of fuel, huge amount of temperature and pressure. But these tiny creatures, the cyanobacteria, fixes atmospheric nitrogen very easily. So it's a very good tool to fix atmospheric nitrogen in the agriculture ecosystem. And also the cyanobacteria fixes 25 to 30 kg of nitrogen per hectare per season. Imagine. So this amount of chemical fertilizer can be reduced when we use cyanobacteria as a tool. In addition to the nitrogen, the cyanobacteria can also secrete different vitamins and hormones, which is useful for the plant growth. And also these cyanobacteria, when it was used as a bioinoculant or biofertilizer, it increased the yield by 10 to 15%. So when the farmer is getting 100 kg by chemical fertilizers, means when he add the cyanobacteria, he can reduce the chemical fertilizer up to 25 kg and the yield can be increased by 15 kg. So it's a very good tool as well as to save our soil health, to save our mother nature. In future, the usage of algae is more important in the agriculture ecosystem. So we have designed a project to isolate stress-tolerant cyanobacterial strains to produce, and we also developed a consortia of the cyanobacteria, and we have evaluated the ability of the nitrogen fixation in terms of nitrogenase activity, because nitrogenase is one of the important enzyme which is involved in the nitrogen fixation, as well as the polysaccharide production, as well as the growth hormone production. And finally, we have evaluated the development of stress-tolerant cyanobacteria in the application in the rice ecosystem, how it improves the plant growth, how it removes the sodicity injury in the plant. Like that, we have evaluated the pot culture method. So these are the different cyanobacterial cultures that we have isolated in different ecosystems. In the Tiruchi soil, because Tiruchi is well known for the sodicity patch, as well as in Tiruchendur, in Thoothukudi seashore, we have isolated all these cyanobacterial cultures. So it can be used to avoid the sodium ion injury to the crop plants. So it can be used as a tool for the remediation for seawater intrusion in and near Thoothukudi, in and near Tiruchendur. So in the agriculture field, when they are using these type of cultures, the plant stress can be minimized. These are the cultures which we have isolated: the nostoc, the Sclopsis, and some of the green algae from the sodicity soil. When we use these consortia of these algae, the sodium injury is minimized for the crop plants. And we have also mass multiplied these cyanobacteria in different sodicity level. So we have artificially created the sodicity level in the laboratory by 2.5 molar sodium carbonate, 5 molar sodium carbonate, and 7.5 molar sodium carbonate, and different level of sodicity was developed different level of sodicity is developed in the laboratory itself in the in vitro experiment, and we have artificially inoculated the cyanobacteria, and we have studied the tolerance, sodicity tolerance, and we have found some promising cyanobacterial cultures which can be used in the field level. And we have also performed a field assay where when we are using the cyanobacteria, Anabina, nostoc, the plant height and the panicle length, as well as the number of fertile panicles, the panicle weight, seed yield, as well as the harvest index has been increased. So these cyanobacteria, this sodicity tolerant cyanobacteria could be a promising tool to for the upcoming seawater intrusion problem or other problem. These are some of the publications which are our recent publication. Like this is a publication which were published in 2021 with a NAS rating of 10, and this is a publication at 2022 at NAS rating of 8, and this is a publication 2022 by NAS rating of 12, and this is 22. We have done the publication at NAS rating 10. So we are always open for collaboration. If anyone is interested to collaborate with us for any projects, these are the projects which I have handled. We have handled DST served projects worth of 25 lakhs in a nano biotechnology program, and we have also developed a PGPR as well as mycorrhizal fungi for a worth of 54 lakhs, and we are having an exclusive biofertilizer production unit, and we are working at the plant microbe interaction, as well as we are having international collaborative projects as well as with Sri Lanka, and we are open for any type of collaboration. If anybody is interested, this is my email ID. You can feel free to contact me through the email for any type of collaboration or publication or any type of project. Thank you.
Thank you, sir. That was a nice presentation.
Thank you.
Audience, now the session is open for discussion. Do you have any questions? You can ask. The audience, do you have any questions? Sir,
Yes, madam.
can I ask you a question?
Please. Yes.
Just a basic question, I think so.
Sure.
So most probably, BGA as a biofertilizer are being used for the high yield of rice plants.
Okay.
Is there any specific reason for using only rice plant, or we can use this for some other plants too?
Madam, as you know, this rice ecosystem is always having enough water. Okay. So, stagnated water system. We are growing the rice ecosystem, rice normally in the flooded ecosystem. So, as algae needs enough water, so the cyanobacteria is commonly recommended as a biofertilizer in the rice ecosystem only. And recently, it's a very good question, because recently for less moisture consuming crops also, the cyanobacteria is recommended as a biofertilizer as a pellets. So when you want to recommend these biofertilizer, algae as a biofertilizer, it needs moisture. Okay. So we are now we are recommending this BGA for rice because it is having enough moisture. So when you have a plant ecosystem with enough moisture, you can well ahead, you can use these cyanobacteria as a bioenzyme. No problem.
Okay, sir. Thank you. Any other questions?
Sir, I have one question.
Sure.
Sir, regularly cyanobacteria in the moss cultivation of cyanobacteria is very difficult. You got the good amount of pigment from the Vetilopsis. How you can get this from Vetilopsis? Any growing media you specially used?
Yes, this is also a very good question, because growing cyanobacteria, purifying cyanobacteria and mass culturing is a type of challenge, and it's a kind of art, I can say, because you used to get many contamination like green algae or other mixture of algae. So we used to have a stereo microscope. We used to purify the culture first, and we have a specific medium. We have a specific medium, as you have told. We have a specific medium which induce the pigment production, as it's a protein. We have a specific ingredient also to increase the pigmentation in the cyanobacteria. And also
some stress to that cyanobacteria. Am I correct, sir?
It is not. It's not stress. We can also add a precursor which increase the protein production in the two way, because when you create the stress, the chlorophyll will be reduced and the phycobilin production will be increased. That can be seen in the cyanobacterial physiology. But when you want to go to the commercial level, you need to add the precursor of phycocyanin production. That will be the good idea. We have identified some of the precursors in terms of nutrients which induce the phycobilin production, as well as a specific medium also. We have a very good amount of culture collection. If anybody is interested, we can have a collaborative project or something.
Sir, another one question. You used one cyanobacteria and the green algae consortia for the improvement of soil.
Yes, madam.
You didn't mention about that green algae name.
Because, madam, we have identified the cyanobacterial cultures, but the green algae, we have showed that picture because it is a presence. Because we in the sodicity ecosystem, the cyanobacteria as well as green algae is present. Like that we have told, but we have not taxonomically classified the green algae. Okay, we have used as a mixture. But as we are already known about the cyanobacteria, we have taxonomically identified that algae. But the green algae we have used as a consortium. We have not identified that culture.
Okay, sir. Thank you. Nice presentation, sir.
Thank you, madam. Thank you. Madam, shall I close my presentation, madam?
Okay, sir. You can leave. Thank you.
Thank you.
Any other presenters who's willing to present in
Ma'am,
today's
I'm
session?
Swati. I'm ready to present, ma'am.
Okay, you can share your screen.
Ma'am, make Rama Prabhavati as a host, ma'am, because I can't able to share my slide from my phone.
Yeah, I have
Please.
made her as a co-host. You can share.
Okay, ma'am. Hello, ma'am.
Yes, you can start.
Am I audible, ma'am?
Yes, you are audible, ma.
Okay. Good afternoon to all. Myself, Swati, PhD scholar from Annamalai University. I'm going to talk about the topic microalgae as a renewable resource for sustainable agriculture. First one is introduction. First, what is microalgae? Microalgae is commonly known as seaweed. It is also known as phytoplankton, microphytes, and planktonic algae. It is a photosynthetic organism because microalgae found in both freshwater and marine water. Also, they form an enormous population in the aquatic ecosystem. Microalgae do not have true leaves, stems, or root like other plants, but they also have chlorophyll pigment that help to trap the sun energy. This help to make photosynthesis. And microalgae is a source of biofuels, bioactive medicinal products, and food supplements. They have an outstanding photosynthetic efficiency and biomass productivity with high content of fatty acid, polysaccharides, and proteins. They have a great potential to be used as a biological resources in the field of medicine, health product, feed fuel, and agriculture. Particularly in agriculture, these organisms have ability to enrich soil nutrients and enhance utilization of macro and micronutrients. And then role of microalgae in agriculture. In agriculture production, different pollutants from wastewater, including pesticide, fertilizers, and rural domestic waste, which contaminate the soil because of the lack of fixed pollution emission point. So, the use of different type of microalgae could purify the different type of wastewater and harvest the microalgae biomass as a fertilizer and greatly reduce the production cost. And then it helped to improve the soil fertility through its own growth, promoting microbial decomposition, improving the transformation and the circulation of soil material. And then it helped to sequester carbon, thus contributing to sustainable agriculture. Because microalgae are capable to absorb CO2 from the atmosphere and sequester in the soil. And this picture shows how microalgae help in the agriculture. First of all, different industries, industrial sector, agriculture sector, and municipal sector, these sectors release CO2. Microalgae capture these released CO2 and then produce the biomass. And these biomass help to produce the biofertilizer. This biofertilizer used as a fertilizer in the crop production, and these crop economic product will be sold in the market. And then uses of microalgae. First of all, microalgae used as a biofertilizer. Biofertilizer containing bacteria or fungi has shown good result for a wide variety of crops, and the use of microalgae has gained strength due to their unique properties when applied to the soil. And then microalgae is a valuable source of organic carbon. This is the key aspect in agriculture. Considering the depletion of soil organic carbon is a significant form of degradation in cropland, leading to decreased soil quality and soil fertility. But microalgae can assimilate organic carbon into their biomass through photosynthesis, and several strains can release exopolysaccharide, which help as a carbon source and carbon sequestration, which enhance the soil aggregation and the soil stabilization. And then microalgae influence soil microbial population that have positive impact on the plant growth, as well as it helps to control the pests and pathogens. And then microalgae improve soil ecological health. Among other soil microbiota, the novel microalgae alone account for about 27% of the total biomass in agricultural land. And then cyanobacteria are more suitable as a soil biological indicator for land use and have the potential to improve the soil health and fertility. Green algae and cyanobacteria produce abundant organic matter during climate change. It increases the soil organic carbon by releasing exopolysaccharides during algal cell decomposition and become a readily available form of carbon required for the growth of soil microbiota. And then microalgae used for wastewater treatment. Use of microalgae for wastewater treatment is more advantageous than compared to the conventional wastewater treatment. First one is economical. It is cost effective for the removal of phosphorus, nitrogen, and pathogens when compared to the sludge process and other secondary treatment procedures. And then low energy requirement. Conventional wastewater treatment process involve aeration, which is energy intensive, whereas algae-based wastewater treatment produce oxygen that is needed for the aerobic bacteria, and algae offers an efficient way for nutrient consumption and provide aerobic bacteria with oxygen through photosynthesis. And then greenhouse gas emission reduction. Conventional wastewater plant contributes significantly to greenhouse gases, but algae-based wastewater treatment release CO2, but the consumption by algae is greater than the release. So, it making the whole system carbon negative. And microalgae as a biofuel. Number of government agencies and companies are funding efforts to minimize operating and capital cost and making algae fuel production commercially viable. And attempts are being made to cultivable algae into large amount for making bioethanol, biodiesel, biogasoline, biomethanol, and biofuels. They can be cultivated within minimal impact on freshwater resources, and it can be produced using wastewater and ocean water. So freshwater not wasted, and biodegradable and harmless to the environment even if spilled. And other uses of microalgae. Algae can be used for trapping fertilizer present in the farm runoff. After the harvesting, they can be used as a fertilizer. Since algae can grow in harsh condition and do not require many nutrients, they are cultivated in places that are not suitable for agriculture purpose. Therefore, they do not compete for the arable land as well as use wastewater, not fresh water. And unlike row crops, algae does not depend on specific seasons. Algae can thrive wherever there are warm temperature and plenty of sunlight. And then, since algae is carbon neutral, it can help the environment by taking CO2 from the air. Algae farms can be located near carbon producing refineries and power plants. Conclusion. Microalgae offers wide range of environmental application that can be contributed to more sustainable future. From biofuel production to wastewater treatment and nutrient cycling, their versatility and efficiency making them a valuable resources. By harnessing the potential of microalgae, we can reduce our dependence on fossil fuels, mitigate climate change, and promote environmental stewardship. Embracing microalgae-based solution can pave the way for a greener and a cleaner world. Thank you.
Nice presentation, Ms. Swati. Can you please show me the first slide? Introduction slide.
Ma'am.
First slide. Title.
Okay, ma'am.
Yeah. The title is clearly written as Microalgae as a renewable resource for sustainable agriculture. But your
Yes.
presentation is mainly based on all the applications of microalgae.
Yes, ma'am. Because microalgae is a renewable resource. After renewable only, these microalgae used for these processes.
But the title is sustainable agriculture.
Yes, ma'am.
But you told about the biofuel and the phycoremediations. That's why I'm asking.
Ma'am, sustainable agriculture means we have to cultivate for the longer period. Microalgae used to produce biofertilizer. This biofertilizer help to improve the soil fertility, and so soil viability
Yes, you
also
are correct.
long.
But your presentation is deviated to other application also. Whenever you present some work, you must concentrate on the single field. That will give you more depth. That's why
Okay,
I'm asking. Okay.
Okay, ma'am. Next time I'll correct it, ma'am.
Okay.
Thank you, ma'am.
Dear audience, do you have any other questions? Swati,
Yes, ma'am.
can I ask you a question?
Ma'am.
And your presentation was quite general, clear. So I appreciate you for that first. I
Thank
just
you, ma'am.
wanted to know, since you are a scholar, what is your research work in this?
Ma'am, I'm not researching this particular topic. I'm just collecting the data from the secondary sources like articles and the websites, ma'am.
Yes, that is what I think ma'am was also trying to say that.
Okay, ma'am.
Since it is a presentation, we just wanted to know what is your contribution in this. We thought it may be your own research work.
No, ma'am, I'm not doing any research in this topic, ma'am.
Okay. Our wishes to you. Thank you, Swati.
Thank you.
But you collected many reviews from many article, I think, so that it deviated and go for the general. Okay, thank you.
Thank you, ma'am.
I invite Ms. G. Rama Prabhavati, research scholar, Department of Agronomy, Annamala University, to present your research work. Ms. Rama?
Yes, ma'am.
Are you ready to present?
Yes, ma'am.
I just want you all to remind that kindly stick on to the time allotted for you. Your presentation time duration is 7 minutes, which is
Okay, ma'am.
followed by 3 minutes of discussion. Kindly stick on to it. You can share your screen. Your screen is visible and you can proceed.
Okay, ma'am. Good afternoon to all. Myself G. Rama Prabhavati, research scholar in the Department of Agronomy from Annamalai University. My topic is about microalgae as a biofertilizer, which is a sustainable way to improve soil fertility and plant growth. The content I cover: introduction, recent trend in agriculture production, role of microalgae as a biofertilizer, advantages and its disadvantages. First, introduction. Microalgae is a photosynthetic organism. It includes eukaryotic green algae and prokaryotic blue algae. It has potential to be used as a biological resources. And these fascinating organisms can be also used in agriculture field, which enhances the soil fertility and improves the soil health. And once the soil fertility is improved, automatically the yield also improved. And next, recent trends in agriculture production. So nowadays we are using chemical fertilizer for getting high yield due to high population. By using high fertilizer for increasing the productivity, the soil fertility is degraded. And then for agriculture purpose, we are using water also. Water is the main source for its growth. The water is nowadays get polluted because of so many industries have been developed. So by for rectifying the water pollution in the water, by using this microalgae can be rectified for long rectified, and it improves the soil fertility. Improves the soil fertility by releasing nutrients. Role of microalgae as a biofertilizer. Microalgae can be used as it can be used as beneficial for soil nutrients recycling, and it promote the plant growth by improving nutrient availability in the soil by producing macro and micronutrients, and also produce bioactive substance such as phytohormones, forming root association, or by protecting plants against phytopathogens and pests. Improves the soil physico chemical properties. Microalgae secretes a variety of active substances such as carotenoids, proteins, fatty acids, and plant hormones, which helps the soil to get more fertile, so that the plant can absorb more nutrients from the soil, and it plays important role in plant growth and development by providing nutrients for soil microorganisms and increase soil organic carbon, and thus affect soil properties, and it improves the soil microbial community. Microalgae has a potential and wide range of increasing soil fertility due to depletion of soil organic carbon. Microalgae convert carbon into sugars through photosynthesis, which also affects soil microbial community structure, and thus improving the aggregate of the soil and its stability. Having this multifunctional activities, especially for cyanobacteria, it can secretes plant hormones and active substance for the control of pathogens and pests. So advantages and disadvantages of microalgae as a microalgae can reproduce quickly, and it promotes the plant growth and development, and the cost of production is very low. They can prevent nutrient loss by slow releasing of nitrogen, phosphorus, and potassium to meet the growth needs of plants. And disadvantages: it is difficult to store because it have a shorter shelf life than chemical fertilizer. And for production of microalgae as a biofertilizer, it requires specific machinery, and very high nitrogen concentration can cause deactivation in the production of pigments needed for photosynthesis. Thank you.
Dear audience, do you have any questions? Dear audience, do you have any questions? Ms. Rama,
Yes, ma'am.
and your topic also was little bit general, but your
Yes,
presentation
ma'am.
was
Because
nice.
I didn't also do research in this topic. I collected general informations about microalgae, which can be
I
used
hope
as
you
a.
have just started your research work, right?
Yes, ma'am.
Have you just started your research work?
Yes, ma'am.
Okay, I appreciate you.
Okay, ma'am.
Congratulations, Rama Parvati.
Yes, ma'am. Thank
It's
you.
very nice presentation. I want to know, do you know any microalgae used as a fertilizer?
Ma'am, blue-green algae, ma'am.
Any name, particular name?
Is cyanobacteria, ma'am.
I want to know about the species name with genera species.
So,
Any generic name is also okay.
Hazola, ma'am.
It's not a microalgae. Azola is a pteridophyte.
Yes, ma'am.
Uh, okay, you collect a lot of review on the microalgae as biofertilizer. So I want to know, you know the name of some of the cyanobacteria used for a biofertilizer. Okay, thank you.
Okay.
Now I call upon Dr. K. Maheshwari to start her presentation.
Good afternoon, ma'am. I couldn't share my screen. Can you please help?
Can you please try now?
Okay, ma'am. Yeah. So, is my screen is visible, ma'am?
Yeah, you can proceed.
Okay. So, good afternoon, everyone. Myself, Dr. Maheshwari from Haryana. I'm here to present my research work, that is Taxa of Desmidaceae from Tamil Nadu, Starodesmus. So, coming to introduction. So desmus is a group of non-metallic organisms. This is exclusively present in fresh waters. And if we see desmus, no, it will be having a thick or thin mucilage that will be held this to entangled as a colony or a solitary. It will be of a solitary form or it will be even as a filamentous form. Even though if it is a filamentous form, if we have slight disturbance, so it will be dissociated into individual cells. In that, so we cannot call this as a true filamentous form. Coming to is nature, it is classified into two types, that is placoderm and sacoderm desmus. So the placoderm, which means true, and sacoderm, which means false. Why it is true and why it is false in sense? If we have a deep construction, basically a desmid is beautifully diverse to form in green algae. And we have a construction at the center of the semicell. There will be two semicell. If there is a construction in that, that will be called as placoderm. And if there is no construction in that, will be called as sacoderm. And the very important point in this group is they exhibit a conjugation mode of sexual reproduction, which is like very pioneer. And that's how it will be called as a highly evolved form among other algae. Moving into the topic of the day. So it is about Staurodesmus. So this is a quite complicated genus. So I would like to take you to know about the history. So it was like Ehrenberg in the year 1838. He actually just grouped every cell which is converged and with the spine that will be called as Arthrodesmus. But after him, Ras 1848, he actually treated that is like the organism or the green alga, which is like monospinous, which means only one spinous, and with be having elongated processes, and it will be having an angular in end view. Can you see this picture? It will be having an angular in end view. If it is in this category, then it will be belong to Starastrum. But when we have the only two spine on the either end of the semicell, then that should be called as orthodesmus. This was a classification, or this was the treatment he given given by Royce in the year 1848. But when coming to tailing, he actually just proposed a new group because it created so much mess and it should be treated differently. So he actually separated a different group as Staurodesmus, naming the combination of Orthodesmus and Starastrum. Can you see Staro from Starastrum and Desmus is from Arthrodesmus? So there are some taxa which will be forming in between these two, that should be treated as a different genus. And so tailing in the year 1948 evolved a separate group for these particular taxa as Staurodesmus. When coming to the diversity, yes, we actually initiated this work in the year 2013, and from 2013 to 2016 we elaborately collected all the freshwater habitats, and I actually concentrated only on diversity of desmids. And in totally out of 32 districts on that time, we actually explored 30, and totally 1432 that is like 1432 samples were collected, and out of 15 different habitats like ponds, lakes, rivers, dams, canals, and all the possible habitats. And out of these many samples, I can harbor 258 taxa of desmids from 26 genera. And today I have taken only Staurodesmus as a genus, and in that Staurodesmus genus, I have 11 taxa to present to you today. So coming to the materials and method, the way we collect algae is like how we can able to, what are the way we can just explore in the freshwater habitats. We just explored in the way, and we actually collected all the submerged aquatic plants with the forceps. And we know the aquatic plants, that is like filamentous alga, that will be harbor more desmids, because desmids are usually lie on the above of the filaments, like it will be periphytic group. So so whenever we can able to collect apart from the water, so it is like we can able to expect a large number of diverse group of desmids. And coming to the preservation of samples, of course we actually preserved in a pet bottles, and we used 4% formalin to concentrate the samples. And these are the some of the pictures to show you about our habitats. So coming to the point, so Staurodesmus, I'll be explaining out of the 11 taxa, I'll be explaining all the taxa with this diagrams, I mean with the colorful pictures and the main key to identify it. So the first one we will be seeing is Staurodesmus unicornis variety gracilis. Can you see, this is an apical view, that is we can see the alga from the top view. And this is a front view. In this front view, we can see this is one semicell, this is one semicell, and this is the middle part that will be called as isthmus. And we can see there's a strongly diverged, it is like we call it is converged. So the isthmus is like widely open. The sinus is widely open. We call this as a sinus, and this part is called as isthmus. And we can see the spine is strongly curved towards on either side. So this defines this taxa, and this was collected from Elixir Lake, and it was, I have actually mentioned all the pH and temperature and where it is collected and the characteristic feature to identify niche. Okay, the next one will be Staurodesmus digictus and Staurodesmus cuspidatus variety divergence. Yeah, if we see, no, if we orally see, it will be looking alike. But if we keep on seeing an in-depth, when we see Staurodesmus digictus, we can see the semicell with the convergent spines over there on the either end. But when we see the cuspidatus var divergence, we can see there is no spine on the apical area of the semicell. Rather, on the lateral area only, we have a strongly convergent spines. And if we see the isthmus area is like very, very short. When we compare with this cuspidatus var divergence, we have very elongated sinus, and this signifies this is differs from this. And moving on to Staurodesmus megacanthus, and this is an angular view of the cell, and this is a front view. And the sinus will be very open, and the spines will be acute. Acute in a sense, the spines on the lateral angle is very sharp, and the semicell will be totally triangular. This genus will be varying from elliptical. The semicells will be varying from elliptical to triangle, and we also have the cunate shape. So that we will see in later. And now it comes to the Staurodesmus brevispina variety obversus, and here it's very short form, okay, and the spine will be like very, very small. It will be totally bowl shaped semicell in very short spine. It is. We can't even find it in the light microscope. It's very very short when compared to the other form of this genus. When come to Staurodesmus convergence variety Laportier, and here we can see the semicell is totally semicircular in shape, whereas we couldn't see prominent spines when compared to the other forms. So we will be, but still we can see some projections over the lateral ends. So we will call that as a papilla instead of spine. So it is like spine-like papilla are present on the either end of the semicell. And now come to the Staurodesmus gibberellus. We can even see now, we can compare with these two forms. The gibberellus will be having a very stout spine, very converged, and it is very short. But when we compare to the Staurodesmus convergence, we couldn't even find the spine. So it is papilla-like. Hope you can understand the difference between these two forms. And coming to the Staurodesmus convergence, and this form you can easily see in many of your collection. It is a commonly available form of this genus. So Staurodesmus convergence, we can see the spine is quite long and it is very sharp, okay, and it's not convergent, it is straight. So if we see convergent, you can see this form, Staurodesmus curvatus variety latus. The spine is totally angular and it is totally converged. It is converged to the end. But if you see Staurodesmus convergence, it is totally sharp. It is not, converged towards the center. And that's the difference between the Staurodesmus convergence and Staurodesmus curvatus variety latus. And comes to Staurodesmus mordex, though we can see, of course, the semicell is elliptic, everything looks similar. But the issue is, we couldn't see any spine over the lateral ends, whereas we can see the wing-like structure. Hello? Yes, ma'am.
Sorry to interrupt. Kindly stick on to the time, ma'am.
Okay, fine. So this is with the Staurodesmus mordex, and when it comes to Staurodesmus corniculatus, it is having a cunate shape and no spines will be present. And these are the new reports to India and Tamil Nadu from this genus. And this is the total or overall picture. And these are my references. And I deeply thank my guide, Dr. Balsami sir, who actually, without him, definitely this stage will not be possible. And I really thank him from bottom of my heart for his strong motivation and guidance. I also thank Dr. Mary Shanti, assistant professor of this college, and thank you so much, Akka, for motivating me to be presenting this topic today. And of course, I thank my families and thank you all.
Thank you, ma'am. That was a nice presentation.
Thank you.
The audience, do you have any questions? Ma'am,
Yes.
Kindly let me know whether you have done this research work on staurodesmus for more than a decade.
Ma'am, I have done for entire desmids. Desmids including like nearly 27 genera that I actually given you one genus today.
Okay.
Yes, ma'am.
The audience, do you have any questions? Thank you, ma'am.
Yeah.
Do you have any other papers to present?
Yes, ma'am. One more paper is there. Shall I?
Are you going to present now?
Yes, ma'am.
Yes, you can proceed with that.
Okay, ma'am. So moving on to the next topic of the day, that is some observation of few taxa from genus Staurodesmus from Tamil Nadu. Of course, this paper totally will be talking about the SEM aspect. And of course, there's an introduction, and I'll just tell you the important points. Though there is a SEM work was done very lightly on this group is concerned, there are like few good papers that will be help me especially to locate and identify when come to the SEM, because like light microscopy is totally different and SEM is different. And when come to desmids, and it is like very tough because it's totally ornamented. And so I am fortunate to get these references like Cotentel in 1981 and Johannes Kermer in 1982 and Anisimova and Stair in 2018. And based on the surface ornamentation only, we actually got these many types. Okay, that is like according to Cotentel, homogeneous smooth, homogeneous corbical, homogeneous smooth and vericate, heterogeneous smooth, scorbical vericate, heterogeneous smooth, scorbical spiny. I will not read all the thing, but I'll just given. So for example, this is how we can able to read it. This is based on Neuhaus and Kiermer reference, and it was published in Gonchrav et al. In the year 2001. We can see this is Staurodesmus wildmania. We can see the pores. I hope you can see the pores are totally scattered. And here there's a Uastrum species. We can see the spores over. Actually, this is called depressions and scorbicle, which is present over the apex. And this is the pore type of 5. We have different types based on him. Okay, he actually given totally 5 types of pores, and it will be totally concentrated on Uastrum when compared to other genus. Okay, and when come to Anisimova, and she also grouped totally 5 classifications based on morphology and distribution of scorbicals on the cell wall and also the ornamentation. And I just given the details over here. Since it is like more than group, 4 groups falls only for genus Uastrum. Like you can see all the ornamentation type in the genus Uastrum. But today I will be talking only about Staurodesmus. And here in desmids, it's very scarcely studied group in India, and that was SEM study only Professor Vidyavati ma'am actually done so far, right from the year 1982 till 1988. And come to the materials and methods, we of course we actually preserved all the field samples in a formalin that was taken, and it was kept washing in the distilled water, and we have taken to the acetone grade series with like 30%, 50%, 70%, 90%, and finally it was preserved at the treated with the 100%, and then it was with the 10 minutes graph. This will be followed by critical point drying, and we will be coated the organism with the gold, and then we finally operated at the SEM with the 5 to 15 kV old. And these are the results. I've just given a few tags of Staurodesmus. This is Staurodesmus percatum. For audience understanding, I just given the light microscope picture and the SEM picture. We can see very great differences between these two. And especially if we see Staurodesmus albans, the light microscopy we couldn't even see the spores. And here we can see in SEM, we can even study how the type of pores is there, what are the type, how it is distributed. And so it will be very important to know the study this complicated group, especially with the help of STEM study. Then comes the Staurodesmus handshake variety congruent. Here you can see the differences. And this is Staurodesmus manfieldi variety splendidum. Here you can see the arms and the scorbicules, how the patterns, and the cell wall over here is very smooth. No pores are present. In that way, you can identify. And then comes Staurodesmus pinnatum variety hydra. This is Pharma supernumerianum. You can see the light microscope view. This is apical view with a lot of process over curved in that way. Here you can see a beautiful SEM picture where the arms with a variety grade of the spines over there. Okay, and this comes Tarishan Johnson. Here we can see the same thing, long processes, arm with the spines. We can see bifurcated spines on the lateral arms. And here the very interesting feature, I actually took very long time to identify this because this is Tarishan ralphsii variety depressum. Here we can see the light microscope study, it will merely look like Cosmerium. It is very, very tough to identify whether this is a Starastrum or Cosmerium. But only the same picture actually revealed me, and we can see beautifully arranged the net-like reticulate pattern on the entire cell. It was like a center. We can see the totally hollow, and if you see the apical view, we can see this 4 scorbicule here. This is one type of 4. This is like another dense reticulate thing, and this is the sparse reticulated thing. So this is a very beautiful picture in my entire collection. And coming to the conclusion, of course, as you know, this is very important to study the SEM parameter because light microscopy will reveal only a few parameters. But if we go for SEM observation, definitely it will be helping for us to crack, especially taxonomic disputes group like desmids. And very, very small cells for Cosmerium, it is like very tough for me to identify. So this actually helped me a lot. And diatoms and desmids are the two groups which is like very beautiful. You can see you can't even take your eye from it when you are observing under the microscope. So these groups definitely need SEM treatment because you will be amazed to see all the structures. And of course, this is documentation is very, very important because it was like no one studied or it will be very, very less. So these are my few references. And of course, again, I would like to thank my guide and Dr. Balasamy sir and everyone else and to support me. And without them, definitely I couldn't present in before you. Thank you.
Dr. Maheshwari ma'am,
Yes, ma'am.
it's a wonderful presentation. Nowadays, morphology work is very less.
Yes,
You
ma'am.
did very, what we say, it's very important for the society also. Identification is also very important nowadays.
Yes.
Most of the people, desmids and diatom, they don't go for this group of algae. So
Yes, ma'am.
I have one doubt how you isolate this pictures are very clear. That's why I'm asking how you isolate a pure culture.
Ma'am, all this given is like from the field only. It's not pure culture. Pure culture pictures. I also raised a pure culture, but it was with 2 strains of Cosmerium, which I didn't given here. But this is like we usually collect the specimen. I mean, alga from the habitat, any freshwater habitat. We will be just fix it with the 4% formalin, and we will just keep the bottle kept undisturbed for a whole day. And after one day, no, all this alga will be sedimented, and it will be leaving only the clear water above. And we will just fork, and we will just take all the water out, and all the sediment will be collected in a 100 ml bottle, PET bottle like that. Then it will be easy for us to get because our work is actually we actually gone to 30 districts, and it was like very big exploration here. And so like it is very tough for us to carry all the 1 liter bottles, no? So we have to fix it. And so, for example, day 1 we are going, we will be collecting all the samples. We will just fix it, and we will keep it undisturbed, and we will go for day 2. Again, the day 2 collections will be fixed in the same way, but we will take the day 1 samples, and we will fork it, and we will just transfer into the 100 ml bottle. We will just bring it back to our college after our field visit. Then we will immediately identify in the light microscopy study. We will do it, and we will have we will take a picture on your own. And we never did any improvisation. I never did for any of my species. It's a natural color what we got from after our study.
Okay, ma'am. Good. Thank you.
Thank you.
Mr. T. Sivalingam, Research Scholar, Department of Agronomy, Annamalai University. Kindly present your work.
Mr. T. Shivalingam, are you there?
Yes, madam.
Are you ready to present your work?
Yes, madam. Yes, madam.
Yes, you can share your screen.
இவனதான் இருக்கிறேன்.
Ms. Soumya, please be ready. Next is your presentation. Shivalingam sir, are you there?
Ma'am, yes, madam.
Can you please share your screen?
Madam, sorry, madam, I can't be able to share that my screen. Some network issues. I need some time, ma'am.
Okay, sir, get ready.
Ms. Soumya, are you ready now?
Yes, ma'am, I can present now, ma'am.
Since Mr. Shivalingam has some issues with his connectivity, can you please proceed with your presentation now?
Yes, ma'am.
Ms. Soumya?
Yes, ma'am.
Ms. Soumya, are you there?
Yes, ma'am, I can present now, ma'am. Ma'am, can I audible?
Kindly let me know who's going to present now, Ms. Soumya or Mr. Shivalingam.
Ma'am, shall I present, ma'am? I'm Soumya.
Miss Soumya, are you ready?
Yes, ma'am.
Okay, you can present.
Okay. Only the host can share there. Can you make me a co-host, ma'am?
Your voice is not clear.
Ma'am, can you make me as co-host, ma'am?
You are.
Ms. Soumya, you're a co-host. You can share your screen.
Okay. Good afternoon, ma'am. Ma'am, can I am audible? Whether I am audible, ma'am? Hello, ma'am. Whether I am audible, ma'am?
You are audible, but no, your voice is not reachable.
Okay, ma'am. Okay. Today's topic is microalgae as a potential in the food and feed system. I am Soumya, PhD research scholar, Department of Agronomy.
Ms. Soumya, sorry to interrupt you. Your voice is
Yes.
not audible.
Okay, ma'am. Okay, ma'am. I am
Not clear actually.
okay, ma'am. I am S. Soumya, PhD research scholar, Department of Agronomy, ma'am. The topic is microalgae as a potential in food and feed systems. Microalgae are the unicellular aquatic microorganisms which cover 50,000 classified species. The notable species are Nostac, Arthrospira, Apanism menon, Chlorella vulgaris, Chlorella pyrenoidosa. The microalgae has a primary metabolites such as protein, carbohydrates, PUFA, polyunsaturated fatty acids, and vitamins. But the health benefits of microalgae are mainly correlated to the presence of the high value secondary metabolites. The primary metabolites are peptides, polysaccharides, vitamins, PUFAs, and the secondary metabolites of bioactive microalgae are pigments and MAA, mycosporin like amino acids, and phycobilly proteins and polysterols. These are having the positive health benefits, like it has anti-hypertensivity, antioxidative, and anti-cancer, anti-diabetic, anti-cholesterol, and anti-aging properties. Specific microalgal glucans can activate the immune system or expert the antioxidant. The importance of algal lipids is based on the PUFA content, and it has anti-inflammatory effects and the modulation of lipid pathways, and it has neuroprotective action. Microalgae peptides also bind or inhibit the cardiovascular diseases and cancer, while carotenoids can also act as potent antioxidants. The beneficial biological activity will depend on the specific microalgae and its chemical constituents. By Angelina et al. 2017. That's the microalgae has the biomass compounds of carbohydrates, fatty acids, pigments, vitamins, proteins, and the application of biomass is in as it can be used as animal feed, and it is used as human food like cookies and pastas. And in our microalgae in feed purpose, in cattle, it increases the weight and it increases the milk content, and omega-3 fatty acid in meat and milk. In fish, it increases the polyunsaturated fatty acid. In goat, it increases the milk content and it increases the lactose content in the milk. In lamb, it increases the omega-3 fatty acid and it increases the weight in the lamb. In poultry, it increases the omega-3 fatty acid in eggs and it increases the poultry weight. In piglets, it also increasing the digestion and reduces the diarrhea. It is quoted by Matthew et al. 2020. It also useful in increasing the biopopulation of honeybees. The nutritional compositions are like protein, carbohydrates, fatty acid, vitamins, pigments, and antioxidants, like primary and secondary metabolites of bioactive compounds of microalgae is increases the producing high biomass yields, make excellent choice for human and animal nutrition and in agriculture. Based on the functional properties of some microalgae species, it is used as food and supplements, so it can be developed for human nutrition. Moreover, because microalgae contains essential nutrients, it can be also used as a biofertilizer by replacing chemical fertilizer. So it reduce the environmental pollution too. Thank you, ma'am. Ma'am, whether I
It's
am
a nice
audible?
presentation.
Okay,
I
ma'am.
appreciate you.
Okay.
But it's a review type of presentation, and this goes general like vast area.
Yes, ma'am.
I need your presentation should be in specific. Then
Okay,
only you can go in depth to the subject.
okay, ma'am.
Okay, thank you.
Thank you, ma'am. Thank you.
Mr. Shivalingam, are you ready? Shivalingam, sir?
Madam, yes, madam.
Can you please share your screen right now?
Yes, madam.
Please proceed. And before that, kindly let me know whether Dr. Gomati, Assistant Professor TNAU, are you here? Dr. Gomati, are you here?
Madam, she is logging in now, madam.
Come again, sir?
Yes, yes.
Miss Miss Bashida Banu, next is your presentation. So kindly get ready.
Okay, ma'am. Yes.
Okay. Shivalingam sir, please share your screen.
Siling sir, are you still have the issues on connectivity?
Yes, madam.
Okay, then Bashida Banu ma'am, are you ready?
Yes, ma'am.
Okay. Okay, ma'am, you can proceed.
Okay, ma'am.
Shivalingam sir, you please get ready with your presentation. Next will be yours.
Okay, madam.
Ma'am, is the screen visible?
Yes, ma'am.
Okay, ma'am. Good evening to one and all. I am Bashida Banu from Research Scholar from Department of Botany, Holy Cross College, Nagarkoil. So, in this presentation, we will see about the unveiling antibacterial efficacy and impact of solvent variation on Acima tetracantha leaf extracts. So, coming to the introduction, in the pursuit of novel antimicrobial agents to combat the excluding global threat of bacterial infections, botanical resources have emerged as a promising reservoir of bioactive compounds. So, the plant Acima tetracantha is deeply rooted in traditional medicinal practices, has garnered attention for its potential antibacterial properties. The choice of solvents in the extraction process is known to significantly influence the composition and the concentration of bioactive compounds which is obtained from the plant materials. So by understanding how different solvents modulate the antibacterial activity of Acima tetracantha leaves, not only contributes to the growing body of knowledge on natural antimicrobial agents, but also holds implications for the development of effective and sustainable therapeutic interventions. So coming to the objectives, the main objective is to determine the antibacterial efficacy of Acima tetracantha leaf extracts against specific bacterial strains, and also to investigate the impact using different solvents on the extraction of bioactive compounds from Acima leaves. And the plant morphology, the plant is a dense evergreen shrub which is reaching about 2 to 3 meter height, and the leaves are simple, pungent, and oval, and it looks like glossy mid to light green in appearance. And the stems are four-sided, and the spines are occurred in fours at the nodes, and these spines acting as protectors. So a wound from the sharp spine is very painful, so it get the name bee sting bush. And the flowers, they are small, and they are greenish to yellow, occurring one to few in the axis, and it produces attractive translucent yellowish to white berries when they get ripe. So there are some numerous ethnobotanical usage in this plant. The root bark, and the leaves are applied internally and externally for the conditions like rheumatism, dropsy, dyspepsia, chronic diarrhea, etc. And the plant also serves as a stimulant tonic in post confinement. And internally, the leaf juice relieves coughs and pulmonary infections. And externally, it acts as eardrops and toothache relief, and it is also very much effective against snake bites, stomach disorders, and it also functions as a uterine tonic. So these are the testing microorganisms, which include Bacillus subtilis, Enterococcus faecalis, Staphylococcus aureus, Salmonella typhi, Pseudomonas aeruginosa, and Escherichia coli. Among them, 3 are gram-positive and 3 were gram-negative. So coming to the methodology, the healthy and mature leaves of Acima were freshly collected, and they were identified by the taxonomy keys, and the fresh leaves were extracted with solvents, namely diethyl ether, ethyl acetate, acetone, benzene, and ethanol. And the antibacterial activity of aqueous and the solvent extracts are mainly determined by disk diffusion method. Here for that, sterile Wattmann filtered disk 6 millimeter were obtained, and the bacterial suspensions which containing the inoculants were prepared. From that, 0.1 microliter each of aqueous and the solvent extracts were applied to the disc, which is placed on incubated plates, and the plates were incubated for 24 hours at 37 degree. From that, zone of inhibition, if any, around the disc were measured. So coming to the results, from this table, among the tested solvent extracts, the ethyl acetate and diethyl ether showed remarkable antibacterial activity, and some least activity were noticed in ethanol extract. And the ethyl acetate, which showed the maximum inhibition of 20 mm against the pathogens such as Staphylococcus aureus, and which is further followed by diethyl ether extract against Pseudomonas aeruginosa. And the ethanol extract also showing the least zone of inhibition against Klebsiella pneumoniae. So this graph clearly shows the antibacterial activity of the Acema leaves using varied solvent extracts. Among them, the ethyl acetate shows the maximum zone of inhibition around 20 mm. So coming to the conclusion, through systematic experimentation with different solvents, we explored the extraction efficiency and the antibacterial activity of the plant's bioactive compounds. And the leaves, which is known for their pungent and glossy appearance, they prove to be the reservoir of antibacterial agents and the dynamic relationship between solvent selection and the antibacterial efficiency. And the identification of these optimum solvents and the elucidation of bioactive compounds, which is contribute a valuable data for future drug development and natural remedy. So it emerges as a promising candidate in the pursuit of sustainable and effective antibacterial agents. Thank you.
Ms. Bashida, that was a nice presentation. Kindly let me know, you have used 6 different bacterial strains, right? How many were positive and how many were negative? Ms. Bashida?
Hello, ma'am.
Can you hear me?
Yes, ma'am. Yes, among them, 3 were gram positive and 3 were gram negative.
Which bacteria, either gram-negative bacteria showed effective results or gram positive?
Yes, which includes Salmonella typhi and Pseudomonas aeruginosa and E. coli. Among them, the maximum is observed in Pseudomonas aeruginosa.
Okay. The audience, do you have any other questions?
Miss Bashida Bano,
Yes,
I have one question. You told that this plant have the antibacterial efficiency. Have you explained which mechanism is help to this plant to have the antibacterial activity? Do you refer?
No, ma'am. I'm just started by using the choice of solvents.
I'm asking about the cell lysis from the secretion of some enzymes.
Yes, ma'am.
Okay.
No, ma'am.
Have you discussed this part in your research work? That's why I'm asking.
No, ma'am. It's a simple mini project like.
Okay, thank you.
Thank you, ma'am.
Banu,
Ma'am.
what is the Tamil name of this plant?
Tamil name, I don't know, ma'am, but common name is bee sting bush.
Okay. Thank you.
Okay, thank you, ma'am.
Thanks for your presentation.
Thank you, ma'am.
Mr. Shivalingam, are you ready? If so, you can share your screen. Sir? Sir, can you please respond? Mr. Shivalingam?
Madam, I am still now issuing, madam.
Hope everyone in today's session have presented except Mr. Shivalingam and Dr. Gomati.
Yes,
Dr. Gomati, are you here?
ma'am, I'm here.
Are you ready to share your screen?
Yes, ma'am. Yes, I can.
Yes, you can share. Ma'am, are you there?
Shivalingam, sir. Shivalingam, sir. Shivalingam, sir, are you wanted to present your research work tomorrow? You consult with the program coordinators.
Mr. Shivalingam, we once again repeat, if you are willing to present your research work tomorrow, kindly consult the coordinators. Thank you. Dr. Gomati?
Madam,
Sir, today you're having issues in presenting, right? That's what
madam,
I'm telling.
I am presenting now, madam.
What, sir? Sir, today you're having issues in sharing your screen. In case if you're willing to present your paper tomorrow, kindly
Yes,
consult the
madam.
coordinators.
But now I am studying, madam.
Now you're ready, but you're not able to share your screen.
Madam, kindly I request, please, coach, host for Swati for screen sharing.
What you want me to co-host Miss Swati? Yes, you can try. I have made her as a co-host. Miss Swati, I have made you a co-host. Can you please help Mr. Shivalingam?
Okay, ma'am.
Okay.
Ms. Komadi, are you sharing the slide?
Yes, ma'am. 2 minutes. Actually, I'm having some issues, so I have sent my PowerPoint to Dr. Sabarinathan. So he's just projecting, sharing.
Ma'am, I'm sharing Sivalingam slide, ma'am.
Yeah. Sivalingam, sir?
Yes, madam.
You present.
Okay, ma'am. Good evening to all. I am T. Sivalingam, research scholar, Department of Agronomy in Faculty of Agriculture, Annamalai University. My presentation topic is microalgae improves the soil health and agricultural productivity. There is a lot of microalgae there. It's mostly the prokaryotic and eukaryotic microalgae are mainly involving the agricultural productions. For example, eukaryotic algae, green algae, and prokaryotic algae. Prokaryotic algae, there is fixing the nitrogen and recycling the nutrients and producing the plant growth hormones. It will help to the improving the agricultural productivity. Due to that abnormal weather conditions and the climatic conditions, the agricultural production will be declined. Under these conditions, the microalgae play the major roles and improving the agricultural productions. It's like act as a soil binders. It will be improving the soil water holding capacity and reducing the water stress under the abnormal weather conditions. And also it's producing the lot of phytohormones, um, um, producing the lot of phytohormones, polysaccharides, and improving the agricultural production, which will be enhancing the soil organic carbon to reducing the nutrient runoff from the agricultural field through the act as binders between soils and plant root systems. There is a three types of microalgae is there. Is cyanospirae, blue-green algae, cyno, chlorophyllaceae, green algae, and bacillophyllaceae diode. These are all major involving the agricultural crop productions. These microalgae mainly involving the increasing the crop yield and also act as the controlling for the pest and disease under the sodic conditions through the eukaryotic microalgae. There is a different kind of microalgae and they are involving the crop improvements or crop productions. There are eukaryotic microorganisms involving the producing the phytohormones and also act as a biocontrol agents and also slow released nutrients. It will be improving the crops. At the same time, the cyanobacteria involving to the nitrogen fixation, phosphorus mobilization through the root nodules, plant roots. It will help to the uptake the more nitrogen, phosphorus, and potassium in the soil. Next. And microalgae also used as a biofertilizer and also microinoculants to the commercial productions and applied to the soil, seeds, or plant surface. It will be enhancing the availability of nutrients for the plant growth. Thank you, madam.
Nice presentation, sir. I have one doubt. You mentioned
madam.
that polysaccharides in the cyanobacteria help for the growth of the plants. Can you
Yes,
tell me
madam.
how this polysaccharide improve the plant growth?
Yes, ma'am. Madam,
You can communicate through Tamil also, sir.
okay, madam. The microorganism involved in the metabolic activity to producing the polysaccharides and phytohormones, it will be help to the plant growth.
Yeah, yeah, already you presented this point. That's why I'm asking how polysaccharide improve the plant growth.
Madam,
Sir,
I don't.
actually polysaccharide give the water retention capacity in the soil, and it increase the soil fertility by the absorbing microbes. The microbes will grow on the polysaccharide and it will increase the soil fertility. That's why. Okay, sir. Thank you. Thank you.
Okay. Thank you, ma'am.
Dr. Gomadi, are you ready?
Yes, ma'am, I'm ready.
Kindly share your screen.
Yes.
For the last presenter of today's session.
Okay.
Madam, kindly enable Sabarinathan to share the slide, madam.
Okay, sir. Sure.
So I have enabled, you can share. Your screen is visible, ma'am. You can share.
Yes, ma'am. Yes. So very good evening to all the members present here. So today's my topic is on probing the functional properties of bioactive compound from spirulina. It's actually a versatile microalgae. Next one. So next slide. Next slide, please.
Ma'am, your voice is not audible, ma'am.
Ma'am, next, I'm just asking for next slide. Okay. So this spirulina, it is a versatile microalgae. It is said to be a cyanobacterium. It is considered as a blue microalgae. It is having the rich composition of bioactive compounds that we are in need of. It is having lot of fatty acids, especially the unsaturated fatty acids and essential amino acids. It is said to be the great protein source for diets. Actually, so many of us are vegan in their diet, so we don't prefer much of animal protein or other kind of proteins. So we were searching for the plant source or something else or derived from the plant metabolites, etc. So this spirulina, it's said to be a vegan protein, and those who want to consume a vegan protein, they can very well go towards the spirulina. It also has the colored compounds like chlorophylls, carotenoids, phycocyanins, phenolic compounds, which are said to be a very good antioxidants. So we all would be knowing the role of antioxidants. So the consumption of a lot of antioxidant fruits or vegetables that gives a lot of energy as well. It just reverse back our aging. So it stops the aging process as well. Next slide. So these are all some of the bioactive compounds from spirulina, said to be the phycobiliprotein, phenolic compounds, and the carotenoids and other chlorophylls. The spirulina, it is said to be a functional food actually. So we all would be knowing the definition of a functional food. These functional foods are nothing than the natural or processed foods that contain adequate non-toxic amount of biologically active compounds. Therefore, it provides more health benefits. It prevents or treats the disease or even their symptoms. So that much effective a functional food is. Not only spirulina, we have a lot of functional foods. So one among the functional food is said to be the spirulina. It is actually a microalgae. It is the emerging source of the functional ingredients called macro and micronutrients. It also has proteins, carbohydrates, phenolic compounds, vitamins, and minerals. Next slide. So these are all some of the biological activities of spirulina. So when we are consuming spirulina, it helps to prevent the cancer. It also acts as an anti-cancer properties. It has the neuroprotective agent. It is said to be a good probiotic. And so there are some prebiotic sources where this probiotic can act. So this spirulina is said to be a very good probiotic one. It has the anti-inflammatory properties. It stimulates the immune system. So a diet which composed of the rich phytochemicals said to be the spirulina compounds, it just avoid the chronic diseases what we are having for a longer period of time, or even the symptoms as well, the neurodegenerative diseases as well, diabetes, cancer. So it is also said to be a good anti-cancer agent. So this is the composition of spirulina, where we could find starting from the water molecule, and it ends from lot of minerals, carbohydrates, proteins, amino acids, fatty acids, etc. Next one. So these are all the some of the macronutrients. So the lipid profile, it's said to be, we have the SFA, that is saturated fatty acids, mono unsaturated fatty acid, and PUFA said to be the polyunsaturated fatty acids. So this PUFA is said to be the potential to be used in specific diets for cases of lipid metabolism disorders. So those who are having some of the metabolism disorders, they are in need of this PUFA, where we could find this PUFA in many of the oils what we are using regularly in our culinary purposes. So one among the macronutrient provider is the spirulina, where we are consuming a lot of PUFA along with the consumption of the spirulina. Apart from that, also the spirulina is having 9 essential amino acids. So these essential amino acids are not synthesized by our body system. So we are in need of this amino acids from the external source. So we couldn't met out all types of this essential amino acids through our regular consumption of plant or the animal food, or whatever we are taking as vegetarian or non-vegetarian. So this spirulina, it provides this essential amino acids for our regular body system, health of the body system. So these are all some of the macronutrients, starting from the protein. So it has 60 to 70% of the protein. So this protein, what we are finding in the spirulina, is not provided in any kind of other sources, not even in the beef or in the any type of meat, whatever we are having. So that much quantity of protein it has. It also has the other carbohydrates and other types of vitamins, starting from vitamin A, K, thiamine, riboflavin, niacin, B6 as well, B12. Next one. These are all some of the minerals: calcium, iron, phosphorus, iodine, magnesium, zinc, selenium, copper, manganese, chromium, potassium, and sodium. So these are all available in milligram as well in the microgram quantities. Next one. Coming to the micronutrients and the bioactive composition. So flavonoids and phenolic acids, they are said to be the phenolic compounds. So the bioactive compounds, it actually encompasses the chlorophyll, carotenoids, phycocyanin, which contributes the major coloring effect of the particular spirulina that is said to be the microalgae. Then the carotenoids, it reduces the damage caused by the exposure to light. So normally carotenoids, they are found in yellow to orange colored vegetables, especially with the carrot. So this spirulina is having the carotenoid content of 25 times greater than our carrot has. Then we have the beta-carotene, then kenxanthin and astaxanthin, lutein, zeaxanthin. These are all some of the main carotenoids. They are found in spirulina. Next one. Next is the other bioactive compound called the phycocyanin. As we all know, this phycocyanin is a blue-colored water-soluble phycobiliprotein. This is important phycobiliprotein such as phycoerythrin and allophycocyanin. So these bioactive substances, they can be used as dyes in the food industry. So mainly it is attractive for, or they are going towards the preparation of infant foods such as candies or some desserts. They are made with this type of phycocyanins. It is obtained from the spirulina. Next one. Next active compound is said to be chlorophyll. So chlorophyll is abundant bioactive compound when compared to other types of colored nature of the spirulina. So it has the essential functions in the photosynthetic process. Apart from that, the green color nature, it actually gives a protection from UV light and other types of environmental or climatic changes that affects the spirulina. Coming to the biomass, the biomass is said to be like it can double its biomass for every 2 to 5 days. So it is actually 20 times more in protein than soybeans, or 40 times that of corn, or 400 times that of beef. Next one. It is said to be a superfood. The reason is it is the role of spirulina, or the usage of spirulina, is nowadays gaining momentum. So day by day, not only in India but also in other types of Western countries, all are using this spirulina for lot of purposes, starting from the making of beverages or jellies or chewing gum, ice cream, cookies, sweets, snacks, or even the pasta or soft drinks, or the dietary supplements via capsules or powders or tablets. So everywhere they are using the bioactive compounds, or the nature of the spirulina powder or the tablet or the capsules, or even a particular compound that is obtained or extracted from spirulina for kind of pharmaceutical purposes, or using the dye, etc., etc. So it is said to be, it is mainly employed in the food industry. So that is why it is called as a superfood. Next one. So we can have the encapsulation of spirulina. This is one important technique called encapsulation, where we can protect the undesirable or the characteristic odor of or taste of the particular algae. Like azolla, the spirulina is also having some odor. So this odor can be avoided when we are consuming it. So there are so many researchers, they have tried towards the encapsulation of spirulina, as well the encapsulation of the particular extracted bioactive compounds. So if they want to extract some bioactive compounds, say for example some carotenoids or some kind of phenolic compounds, or some kind of compounds that is responsible for the medical industry or something, so they are just extracting and doing the encapsulation process. So it increases the stability of the particular bioactive compound. So whenever we are going towards a encapsulation, whether it is micro encapsulation or a nano encapsulation process, it enhances the stability of the bioactive compounds. It also promotes the delivery of active ingredients. So it directly give away the active ingredients in the delivery site. It prevents the degradation due to external factors, say for example temperature or pH or whatever that is inside the stomach or in the outside environment. It used to degrade. So the degradation process can be avoided with the help of the encapsulation. It also improves the solubility, and it facilitates the incorporation of bioactive compounds into systems or any kind of food products. Next one. So there are some biological effects of spirulina species with regard to the particular bioactive compounds. So they are said to be the rich prebiotic as well probiotic. So they can act as both. So these prebiotics are nothing but a substance where the probiotics can act. So it being rich in oligosaccharides, it is also acting as a prebiotic. So it also improves the immune system and it also enhances the activity towards anti-tumor activity. It is the main reason behind this is it is having the antioxidant activity where it can nullify the free radicals. So these free radicals, they are responsible for any kind of disease or any kind of aging process, etc. So to nullify these free radicals, we are in need of antioxidant enzymes. So this spirulina, it is having the antioxidant activity, thereby it is improving the immune system as well. It is having its own anti-tumor activity as well. Then it is also protecting the neuron, like neuroprotective effects. So it just slowing the progression of multiple sclerosis. And it has the effect of metabolism disorders like diabetes and osteoporosis. So there is a particular protein called osteocalcin. So it can be enhanced by the regular consumption of the spirulina. And it also has the anti-inflammatory properties. So the selenium and the spirulina platensis, it is used as a growth promoters, antioxidants and immunostimulants and antimicrobial compounds as well. So this is the overall picture in which it can be used as an immunostimulant. So they have used mice or rat as the animal model, even the guinea pigs as the animal model. So where they are providing this spirulina and it is the output. And coming to the benefits, it reduces the blood cholesterol. It helps to control the diabetes. It also helps in kidney detoxification. It also useful to combating the malnutrition, preventing vitamin A deficiency, and it aids in prevention of anemia. Next one. So this is the one of the research what we did in our laboratory to enhance the protein and carbohydrate content of the spirulina through different substrates. So normally we used to have the CFTRA medium or BG11 medium or Zarux medium for the growth of the spirulina. So we have cultivated this spirulina in different pH along with the different types of medium. Along with that, we have used the generally available organic substrates, whether these substrates could enhance the quality and quantity of protein and carbohydrates. What we used is the cow dung liquid as well the other types of oil cake material and the neem cake extract. And in that we found the cow dung extract, it enhances the protein quality as well the carbohydrate content of the spirulina in the pH of 7.5. So we employed this technique with different temperatures, different pH, different substrates, etc. And we come to a conclusion and we published this paper also. Next one. So this is the RDA, that is the recommended dietary allowance of protein, what we as a woman of 55 kg is required. So this energy can be easily met out by the consumption of spirulina. Yes, thank you. Thank you so much for the opportunity given.
Thank you, ma'am. Kindly let me know the data which you provided. Nearly 3 tabular columns you have given. Maybe if I'm right, I think you have given the mineral composition. Is
Yes.
that your original research work done?
No. It is not the research work done. It is from the research paper.
Okay, ma'am. Okay. Your audience, do you have any questions?
Good evening, ma'am. Nice presentation, ma'am.
Yes, ma'am.
Already WHO announced that it is a secured food. Then why you presented here, ma'am? It seems that all these are taken from the review.
Which one, ma'am?
Ma'am, your presentation, it's
Yes.
total general and the review work. What is your contribution in this slides, ma'am?
Actually, ma'am, we did research on the growth of spirulina to enhance the compound like the chlorophyll, then the carbohydrate, protein content of spirulina.
Ma'am, please show the next slide. I mean, the macronutrients, minerals.
Next
Can you
slide.
please skip the slide? This table only you mentioned that it has that you have taken this from some other research paper, right?
Yes, ma'am. Yes.
Ma'am, it's my humble suggestion to you.
Okay.
In case if you are taking some data from some other paper, I request you to quote the citation inside the slide.
Yes. Yes, ma'am. Yes.
In your future
Yes,
work,
covered.
okay?
Okay. Okay.
It's
Yes.
my
Sure.
humble
Sure.
suggestion to you.
Yes. Yes. Sure. 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 Actually,
Thank
I
you,
didn't
ma'am.
mention. Okay. Okay.
Thank you, ma'am.
Okay, ma'am.
Good evening to all. As of in today's session, there were 10 paper presentations, but 8 out of 10 have presented their papers and 9 papers have been presented. One presenter has presented 2 papers. As we come to a conclusion, I feel really great and thank you each one of you in this floor for being here. So once again, I thank you for joining us today in this international conference on microalgae, biodiversity and bioprospecting. Thank you.
I once again thank you for
Yes, ma'am.
each and everyone for your contribution in this seminar. Thank you.
Excuse me, ma'am.
Yes, sir.
Can we able to see the offline presentations or no?
Sir, online presentation, you cannot view it now, sir. They are just recording it.
Okay. So how we
That's
can
taking
able to
place in
see
some
it
other
later?
hall, sir.
Will be uploading in YouTube or you will be sending it?
No, we'll let you know, sir.
Yeah, sure. Thank you.
So this information will be shared in the group, sir.
Okay, ma'am. Thank you.
Okay, thank you very much, sir. Thank you all.